So this is today's assignment, 510, number 2, transcription activity. And one of the requests made last week was that I spend more time explaining how to actually do the work, which is what we are going to do exactly right now. So in this activity, this transcription virtual activity, there are different types of slides. So slides number 1, 2, 3, 4, and 5 are all the same type of slide. They are an information followed by question slide. So literally read the information and then answer the question or fill in the blanks. So let's do this one together. Human DNA totals over 6 feet in length and contains more than 3 billion base pairs. These base pairs are arranged in specific sequences called genes. Humans are estimated to have between 20,000 and 25,000 genes scattered throughout their DNA. Each one of these genes determines a trait that can be expressed, such as eye color, height, and enzyme composition. In order for a cell to express these traits, it must take the instructions from the DNA and use them to make a protein. This process is called protein synthesis. All right, so I have read that information. So now I have 3 questions to answer about that information on that slide. So slide number 1, or question number 1, says blank are specific base sequences found in DNA that control traits. These are genes. Number 2 asks, give one example of a trait that a gene controls. Now, you could technically write a lot of different things here, but I'm going to use one from the text. And in the text, it mentioned eye color, height, and enzyme composition. I'm going to just use eye color. Now, you could use any of those or anything else, but that's what I'm going to choose to use. Number 3, blank is the process of creating proteins from DNA instructions. This is called protein synthesis. So this is how this type of slide works. You read the information and you answer the questions that follow. You will do the same exact thing for slide number 2, 3, 4, and 5. Any questions on how to complete slides numbers 1 through 5, or anything from questions 1 through question 14. Hearing no questions, we move on. Slide number 6. Slide number 6 is a Venn diagram. Directions. Place each card from the pile below to where it belongs on the I'm reading the directions right here. To where it belongs on the table. If you don't know where to put the card, move it below to the pile and come back to it. All right, so I read the word deoxyribose. And I know that deoxyribose is the sugar in DNA. So I'm going to pick this up and I am going to drag it over to the DNA column. So this is a Venn diagram. Venn diagrams are used to compare and contrast what is unique and what are shared characteristics. The next copy says, copy of a DNA's instruction. I know from my notes last week that that is what RNA is. Awesome. So I'm going to go ahead and drop that there in the RNA column. I don't know what ribose is. I'm going to come back to it. Ooh, double-stranded, no idea. Single-stranded, no idea. Uracil, nope, no idea. Then I get to cytosine. Oh my goodness, I remember in my notes, I saw that cytosine is both in DNA and in RNA. So that's going to go in the both column. So anything that applies both to DNA and RNA goes in the both column. If it is a characteristic only of RNA, it goes in the RNA column. If it's a characteristic only of DNA, it goes in the DNA column. Now, of course, I knew what these were. I was just waiting for a both to come up. Any questions on how to complete slide 6? I don't see anything in the chat. I don't hear anybody unmuting themselves, so we move forward. Slide number 7. This really has nothing for you to do on it. There's no questions, there's no drag and drops. This is just information. So, this is going to tell you all about messenger RNA or mRNA. It is going to tell you about transfer RNA or tRNA. And it's going to tell you all about ribosomal RNA or rRNA. This is information that you might find useful in future slides. Slides number 8, 9, and 10 all work like slides 1 through 5. You read the information and you answer the questions that follow. Okay? Any questions on how to complete 8, 9, or 10? Hearing none, we move on. Slide number 11. Slide number 11 is asking you to construct a strand of mRNA just like the RNA polymerase would do. Drag your nucleotide to the newly forming strand after the promoter. Remember that these bases must be complementary to each other. The first nucleotide has been placed for you. So my DNA template has a C, and my mRNA, the one that I did for you, is G, because C and G complement each other. They are base pairs. Then I have a G, so I would drag over a C. And then I have a C, so I would drag over a G, etc., and so forth. Please complete the remainder of your mRNA molecule. Any questions on what to do with slide number 11? Okay. Slide 12 is also a drag and drop, but we are going to be labeling. So, cards used for labeling are right here. And now I need to go ahead and drag and drop to where they belong. So, for example, my first one says mRNA. And as I look at mRNA, I'm thinking it might be this strand right there. All right. And as you go through, you're going to grab these and you're going to take them to wherever they would go to diagram or to label this diagram correctly. Questions? Hearing none. Nothing's in the chat. Labels, slides 13, 14, and 15 are all like before. You're going to read information and then you are going to answer questions. Now, pay close attention to slide 14 and 15, okay? Because slide 14 and 15 are all about DNA editing, okay? You can even, I think 13 is also included in that. No. So 14 and 15 are all about DNA editing. Come on. Why is it so slow? Oh, there it goes. That's important because we're going to need that information for slide number 16. Slide number 16 says, below is a strand of mRNA that has just finished being transcribed. Remember, transcription is the process by which we are going to create a copy of the DNA sequence for the gene. We are calling that, making the mRNA the original transcription. Now, we have to edit the mRNA so that it's ready for translation, which is step 2 of protein synthesis. Remember that only certain sections of the mRNA strand are used. Also, don't forget to add a cap and a tail. Drag the necessary parts and arrange them into a finished mRNA strand in the box below. Refer back to the previous 2 slides as a guide to mRNA editing. Okay, so I am not going to do this one with you because, again, the information can be found right here. You are good. You will be fine. You got this. And then number 17, these are review questions. So there's no text for you to read, but it is asking you to recall some information from the assignment. Are there any issues or questions about how to complete any of these slides.